So really, this is what I'm going to do. Sort of the do's and don'ts and some lessons learned from our own Mesocosm experience. And I'll try my best to keep within the 20 minutes here. And this is the SAINT acronym that I just talked about. And I'll talk a little bit more about SAINT right now, actually. I sort of have to put this in. It's 5 institutions. It's actually closer to 6, about $30 million from our National Science Foundation. It's a 10-year project, and we're just now starting year 8. So we're pretty mature, and we've run 2 or 3 Mesocosm studies already, and I'll show you some of the results from that. 43 different faculty are involved, lots of students and postdocs. So it's kind of like one of these Framework 7 projects where you have multiple partners, although we try to stick to minimal, but you have a lot of people working within this. What makes this center interesting is that we're very interdisciplinary. So we've got, I'm an environmental engineer, that's my background, so I always put this first, but there's geochemists, there's ecologists, toxicologists, public policy people. We're all working together, and the main goal here is to understand what properties of materials affect their behaviors in the mesocosms, and to collect data that actually has some regulatory action associated with them. All right, so one of the key outputs, I think, of our center has been studying nanomaterials in complex matrices. We don't just use a beaker in our laboratory. We actually go out and we put things in our wetlands. We have some more terrestrial kind of mesocosms. We work with individual organisms. And we have taken a number of different nanoparticle types, put them into these different scenarios, and started to understand how they transform and change in the environment. And Ralph talked about X-ray absorption spectroscopy. That's one of the things that my group does to try and understand speciation in these kinds of settings. And actually, a few years back, we did this paper. It's a feature article, and it just talks about the transformations of nanomaterials in the environment. And I always preface that as that at the time with this paper, all we did was summarize what everybody already knew, but we put it in one convenient place for you to look at. And I'll say it's 4 years later, almost 5 years later, since we really wrote that thing, and most of the concepts in there still hold true, right? They're the basic transformations to think about. And we know once the materials get transformed, once they change in these systems, their properties change. And it's the new properties of that material that control interactions with organisms, control the potential effects. So how do we actually do this? Well, we do work all the way up here at this large scale and with this full complexity, essentially. And I'm going to we'll say a lot more about what these boxes really are in a minute. But we also work way down here. So I do some work looking at where every single atom is in a structure, right? Because that tells you something about how the nanomaterial, what its features are, what its properties may be. And, you'd like to say if something changes at this level, it has an impact up here. And it's not that easy to do. And you have to be working at this scale, but you also have to be working at these very large scales and very complex scales to really understand the behaviors of these materials. And let me give you an example of what I mean by that. Or I will in a second. When we all started this, here in Europe, also in the US, we went in very naively. And we thought to ourselves, all right, I'm going to take a nanomaterial, and I'm going to get some property, like its size or its coating. And I'm going to then map that right onto the effects that it has in the environment. But it turns out that that doesn't really work, because the environment's pretty complicated, right? The environment, the real world, is not our beaker in our laboratory. And the environmental system properties matter, and I would argue more than the initial nanoparticle properties matter for the behaviors of these things, because it affects how they transform, it affects what products are formed. The presence of co-contaminants in an environmental system can have a big impact on their behavior in the long run. And it's both of these that we have to consider when we look at effects. And I would argue the only way to actually measure the effects in the real world is if you include all the complexity of an environmental system. This is what a mesocosm does. This is why we use mesocosms. You cannot study complexity in a system outside of the system. You have to study it inside of the system. Otherwise, there's no way to decipher what's going on. So here's just an example of how a system matters a lot. So this was some work that we did. Ben Coleman is a postdoc. Well, he just got a faculty position in Montana, so he's moving on. But he's done a lot of work with our mesocosms. This was one where we dosed silver into our mesocosm, and all this does is track the organic carbon in the water column here in the mesocosm versus time after dosing. So it's kind of, and you know, there's a bunch of time before we dose, and it's all kind of bounces around at this low level. So you have this stable environment with a certain amount of organic carbon. You then dose your silver nanoparticles. And what happens? Well, the organic carbon goes way up. But we all know that organic carbon interacts with the nanoparticles, right? And that changes the behavior of the nanoparticles, which then changes their potential effects on the system. And so the initial system response is actually changing the particle, which then changes the response on the system. So there's this dynamic feedback loop that's complex and within the system. And actually, if you look closely at this, it's pretty obvious what's happening. We put not that much silver in these systems, but plants don't like silver at all. So here's our control system, a box. Here's silver nitrate. It's sort of hard to see with this light, but it's kind of brown. A lot of those plants died, and that's what releases all this organic carbon into the system. So there's the feedback loop. Now, a combination of a mesocosm, we observe some very different behaviors between two particle types. One particle type was a PVP-coated silver, and the other was a gum arabic-coated silver. And we sort of expected them to behave similarly, but they caused different problems. And this is described in a couple of these papers here from a few years back. But the idea is that the particles go in, they interact with the plants release these exudates, release all the material. The gum arabic ones interact with the exudates, they oxidize more, they dissolve more silver, and it creates this cycle that causes these to be quite toxic. Whereas these particles don't interact with the exudates, they're not enhancing dissolution, and even though they're very stable in the water column, more stable with the plant matter, they aren't having as big of an effect. Because again, the organic matter here interacts with that particle, dissolves more silver, makes it more toxic. Here it interacts with this particle and it dissolves, it doesn't dissolve more, and it actually stabilizes the material. Right? So the moral of the story here is the system complexity, we never would have got this in our laboratory. We had to see it in a mesocosm experiment. And then we went back in the laboratory and did a whole bunch of studies with and without plants to show that this is what was going on here. Again, it's a really nice story. Jason Unrein is a faculty member in our center, does fantastic work. It's worth reading those papers to see the story. All right, so now let me tell you a little bit about our mesocosms. I'm going to just walk through the actual mesocosms that we use in our study. So in about 2006, we started writing this proposal and we had this vision for this facility. And this is the Duke Forest. It's at Duke University. It's in the middle of nowhere. Well, it's near the campus, but it's out. And that's what we had to work with. No infrastructure really at all. And we scratched our heads, once we got our funding, we scratched our heads, wow, how are we going to build our mesocosms? What are we going to do? So the first thing we did is we just dug a big hole in the ground. Actually, a series of them. There are several of them in here. And we thought, well, it's cheap. We don't have that much money we had $50,000 to do our mesocosm facility. That was all that was in the budget. Big mistake. You need more money than that. Which we ended up getting. But this is where we started. The problems here I mean, there are numerous. This is hydrologically connected, so you fill them up, they go, in the summer the water just infiltrates and it goes away. So you can't even keep these full. We tried putting liners in, we tried all kinds of stuff here. This is too simple and it just doesn't work for us. Right? Plus all we have here is water. We don't have any terrestrial side of things. So after a lot of time, and Kurt Richardson is actually a well-known plant or ecologist, and he studies mesocosms. That's all he does. He's part of our team, and he proposed this kind of mesocosm. So we have these boxes. The dimensions are about 3 meters by 1 meter by 0.8 meters, and what it has is a board that sort of slants through the mesocosm. Okay. So you got this board in here at this angle. And what we've done is we filled it up with soil and filled it up with water. And what you get is, in a system like this, is a terrestrial component and you get an aquatic component. And so here you get subaquatic sediments, here you get terrestrial plants. And what's interesting is when it rains, it fills up a little bit. And when it stays dry, it drops down. So you get, just like a natural wetland, you get this transition zone of redox, right? Low redox here, it's oxidizing here, and as the water column goes up and down, you change the redox cycle, and you get some interesting behaviors of the nanomaterials. So it's actually a pretty clever design. So we went and we built these things. So here shows the box itself. We put this food grade HDPE liner in. You know, you get to, graduate students get to get these play around with these things like front loading tractors. They love that stuff. Actually, there was, I should say, not graduate students, it was a postdoc. But he enjoyed driving the truck. And you know, we filled this thing up with our soil, we planted it. This shows some of the plants in the terrestrial side, and then you have your water column. These are just PVC pipes. They're to hold instruments and instrumentation. Okay? So we ended up building 30 of these. You can see all these boxes here. And this is sort of a close-up. And I think, Lars, you were showing a real wetland. And I looked at it and I said, wow, that looks just like our wetland. Right? You got this terrestrial side, the water side. So it's fairly realistic, we think. And then, you know, the students, lots of students get to go out and they get to work in this thing and take samples. So it's a great learning experience for the students as well. Now, one of the lessons that we got from this is at Duke, well, it probably doesn't rain as much as here, but it rains a fair amount. And when it rains a lot. These boxes don't have a drain because we needed to keep them contained. So these boxes were sort of filling up with water sometimes, and we'd have to run out there and pump all the water out so they didn't spill over. And there's a whole process at the beginning of actually taking water from this one and moving it to this one and moving it to this one to homogenize all the boxes because you want to keep them exactly the same. We'll talk a little bit about process in a minute. But they were filling up. And then one winter, it got really cold and they froze. And when you have a bunch of pH probes and redox probes in there and the water freezes, you've lost a lot of equipment and it's very expensive to fix. So we ended up putting a coat. We ended up putting a coat over the top of these things. So they're less natural, if you will. You know, so you can see here, if you look at this whole system now, we have more than $50,000 in this system now. But it's a really nice system. We can control the temperature to keep them from freezing. We do not put the lid on most of the time because sunlight is a big part of the processing that goes on in these things. So we only put the lid up when it's the rainy season or when things look bad, and we can take it on and off as we want. Yeah?
We use quite a similar design, but what I feel is that it sort of might be like a perfect environment for bacteria to grow. As from the first results we get, I'm not sure if we're like, because the humidity in these systems is much higher than in the natural systems. So this is, yeah, might be kind of an issue.
I mean, only when the lid is on.
Yes.
Yeah. So again, we only put the lid on when it's raining enough where we don't want the boxes to flood, or when it gets really cold. Which at Duke in North Carolina, it doesn't get that cold. It might be a week of really cold weather sometimes where we have to do this. So the lid, the top is off most of the time. This is just a cloth that goes over the top, and we just take it back off.
Yeah, we don't have such a thing as a dry season. So.
Oh, okay. Yeah. So it really only rains a couple months a year, and we only do it when we know it's going to rain a lot for an extended period. Then we'll put the lid up. Otherwise, we will let natural rain fall in the boxes. We just only do this when we know it's going to spill over.
Okay.
That's another thing I didn't mention here is frogs. So we don't have a permit to look at frogs. But you know what? When you have these boxes like this, frogs somehow get in there. Even we put a sign up that said no frogs. And they still got in. They just don't, I guess they don't read. It's North Carolina.
You have to put in a little ladder for them to climb up.
The ladder to get back out.
Yes.
Yes.
Yeah,
And
it's something that exists actually.
really?
Frog ladders. Yes,
Wow.
we have them in systems, in large scale systems.
Yeah.
Frog ladders.
Nice. So I'm not an ecologist. I'm an environmental engineer, and I do chemistry mostly. So I get to learn a lot from the ecologists. So I'll tell them about the frog ladder. They probably know
I'll
this.
send you a picture.
The biggest problem is the frogs don't go into all the boxes. Some boxes have frogs and some don't. So there's issues with replication across your mesocosms because frogs actually stir up sediment and pollywogs or tadpoles whatever you call them, that swim around, they affect the system a little bit. So reproducibility is a big deal in these boxes. And if you only have 3 of them per treatment, 3 replicates, and 1 replicate goes wrong, you're sort of on the edge. We talked about statistics. You're really on the edge of where you can do statistics in these systems. All right, so let's think about mesocosms though. Mesocosms are a really, I call it a low throughput assay. We hear a lot about high throughput screening, right? Everyone wants to do high throughput screening of nanomaterials. This is low. In fact, this is, I should say, ultra low throughput screening for mesocosm. I mean, think about each box, right? It's 3.6 cubic meters. There's a cubic meter of soil, somewhere between 400 and 800 liters of water. At the end, we have 10 to 20 kilograms of plant biomass that we have to collect and analyze in each box. And we have 30 boxes. So this is a lot of material. Lots of different fish and insects, right? And they're not the same in each box. So there's kinds of problems with mesocosm experiments that you have to just deal with statistics mostly. And it's a challenge. But it has to be done, because you can't do complex systems without this. All right, so our first mesocosm paper, this, I didn't put the reference here, but it's a 2012 paper. We talked a little bit about not having enough long-term studies. Well, this is a long-term study. It's 18 months, if that counts as long-term. We asked in this study, so I was on sabbatical, so I actually wrote this paper myself. It was nice. I haven't written a paper in a while. It's a very much an environmental engineering viewpoint. It doesn't have a lot of ecology or even toxicology. We asked 2 simple questions. Where did those nanoparticles go in the box, and how did they change? Very, very simple, because you can't answer or ask high-level scientific questions in a mesocosm. You really can't. You have to ask very low-level questions because it's so complicated, you can't do everything. Doing fundamental basic science in a mesocosm doesn't work. You have to do kind of applied science in these boxes. But you can't answer these kinds of questions of where they went and how they changed. So for where they go, mind you, this was just 2 boxes. This particular one was our test run. It was 2 boxes. So we wanted to ask ourselves where the nanomaterials went. Okay, so here's my box. Again, I got water column, I got soil, water column, soil. I went in, I took a sample, I took a column sample, and I did this grid pattern. And then I took my columns and I analyzed 4 different levels, right? So now I've got, whatever this is, 18 samples times 4 levels. And then I have to do replicates. I have to acid digest them all, analyze them with ICPS for total silver in this particular case. So a single box, just to know where it is in the sediment, is an incredible number of samples and a huge commitment. Okay? So the data sort of ends up looking like this, but for one box you have 90 microwave digested samples that you have to analyze. This takes a long time. This is not an afternoon in the lab. This is pretty serious commitment by a person. Okay? Again, if you want the details of where the stuff went, the science of this talk, I'm happy to talk about that with you, or you can read the paper. And that was just one set of measurements to know where they went. In our mesocosms now, we take lots of counts, right? Or lots of things. So we look at prokaryotes, we look at phytoplankton, dissolved oxygen, CO2, methane. All of these things we measure in a box, times 30 boxes, times all of these points, and that's only the first 28 days. And we ran these for over a year. It's an incredible amount of time and commitment, and there's a lot of data and samples to collect. Okay? And then when you finish the experiment, you go and you collect all the plants. That's a lot of biomass. We mobilize about 15 or 20 PhD students and 2 full-time staff people for a month, collecting all the samples and dicing them up and just getting them ready to analyze. Then we have to analyze them. So think of it again, ultra low throughput screening. Okay? Now we don't just have this kind of mesocosm, we have these terrestrial mesocosms that we've worked in. We have a nice stream mesocosm at Baylor University. It's a riffle stream. So up here, it's very shallow, the water's moving pretty fast, and then there's all these little 2-inch tiles on the bottom, and all this periphyton grows, and we can pull out the periphyton from each tile, and we can look and see what it's doing. So think about how big this is. I can't remember how long it is. It's something like 20 meters long. And then each one of those is about 4 centimeters squared. So think about how many tiles there are that one could pick up and analyze the paraphyton on. It's a lot of samples. Now, another main point that I want to make is putting the materials into the system. The dosing of your mesocosm makes a big difference in how things work. So you can have a pulse input where you just put something in and then it stays at that level, if you will. The mass in the system stays at the same point. So you basically put it all in at the beginning, pulse. Okay? The other way to do it is to add a little bit, a lower concentration over a long period of time so it slowly builds up. Okay? So we've done this in our mesocosms and you get very different responses. So our pulse input, this is time, and this is how much silver is in the water column, I think. Yeah, it's water column silver. So here's my pulse. I put a whole bunch in. It stayed in the water column over some time, 28 days, and then sort of leveled out. This is a log scale. It leveled out a little bit. Okay? But if I do the pulse input, low concentration, and I'm pulsing weekly, then I get this kind of sawtooth behavior. Comes down, pulse, down. I eventually end up with more in the water column in my pulse scenario. Oh, really? How much time do we have? All right. I have more in my water column in my pulse scenario than, or in my my press or my low dose scenario than my other pulse scenario. So it makes a big difference. I won't worry about that. It also makes a difference in the response. So here is my low dosing. This is methane concentration that we measure in the mesocosm over time. This is putting it all, or putting small amounts in over time. This is my pulse. I got this big spike of methane. That's because more silver killed more plants, released organic matter, the redox went down, methanogenesis came up. So you get a big difference just depending on how you put stuff into the system. Same idea in our stream mesocosm. When we pulsed, we got a spike, it aggregated, dropped down. If we did a small amounts over time, eventually the small amounts over time led to much more nanomaterial in the water column compared to the paraphyton on the bottom of the system. And that led to very different distributions of mass. Right? So here's my pulse addition. This shows where all the cerium is. So in the pulse addition, all the cerium ended up down here. In the press addition, it sort of ended up more distributed throughout the system. So it matters how you put the materials into the system. Now, another lesson that we learned, we try to do modeling. We try to model our systems. We try to model the sediment. So this is just, I won't go into the details of the model. But the main point is, if you model sediment diagenesis and you put something like silver chemistry into that model, you need parameters for that model. And it turns out this particular model has 38 different parameters that can go into it. 34 of them are kind of set for a system. We know what they are because we've done environmental modeling for a long time. 4 of them are very specific to silver. So like this is the rate of oxidation of sulfur in the silver sulfide. Where do I get a value for that parameter? I can't get it in the lab. I mean, I could try and measure it in the lab. Ralph measures these things with ozone and stuff. But what is the rate of that oxidation in the actual sediment in my system? We don't know. But we would like to measure it in a mesocosm so we could actually parameterize this. So the main point I'm trying to make here is that if you develop models and you sort of know how you want to model your system, then you can make sure that your mesocosm is measuring the things so that you can parameterize your models. So they have to go hand in hand. The modeling and the mesocosm work should go hand in hand because they inform each other. All right, I'm almost done. So this is our workflow, if you will, or design process, and we're just going through this right now for some new mesocosms that we're going to set up in the next month. What I would say, this is how I would go about doing it. First, you want as a group to agree on a conceptual model for your system. What is the conceptual model, how the nanomaterials are going to behave, and you want to agree on what are the key questions that you're trying to address. And you'll find that the different stakeholders have very different questions that they want answered, and that requires different kinds of measurements. And a lot of the stakeholders come in with questions that can't be answered in a mesocosm. They can only be answered in a very well-controlled laboratory system. So to make sure your questions are appropriate for your system and that everyone in the group agrees on what those are. okay? And at this point, you can even plan what are the key papers that I want out of these things and agree as a group that that's the papers that are going to come out. Then you have to acquire and characterize the materials. By the way, this can take like, I don't know, 4 or 5 months to really get right. This can take several months if you have to make the nanomaterial. It might take a really long time. Once you have the materials, you have to determine what you're going to actually measure in the mesocosm and who's going to measure it and how you're going to measure it. So we heard a lot about the measurements, but you've really got to figure out how to do that. And then you also have to figure out how to share your data with each other. A lot of times someone will measure something and then there's no way to really port it or share it across. Right? Then you have to analyze the data. I would argue that as a group, whoever's in charge of different things, you have to analyze it immediately. Analyze it when it comes off and share it right away, because that informs what someone's going to do next. Right? It's a group effort. And then lastly, I just play nice, share your data, and write the papers as a group. You have to be able to. Sometimes people say, well, that's my data. I would like to publish it first before I give it to you. That doesn't work if you're doing a big mesocosm study with multiple stakeholders. There has to be agreement amongst the group that it's going to be joint papers and that it's a collaborative effort. All right, so the main points. Mesocosms are the only way to study complex system response, but they cost a lot, both money and time. You have to do your homework and understand what you want to measure and how you want to measure it. A ton of samples to analyze, a lot of variability, so you can't control everything. Dosing matters, variations that you need to deal with statistics. And I would argue that we want to coordinate our models and our measurements. And I'll stop, and hopefully we've left some time for questions.
Sure.